Hi, first of all, thank you for showing an interest in my work. My name is Gerald Drenten and I will talk about my work titled Exploring Microstructural Changes in Patients with Vascular Cognitive Impairment using multi-B value diffusion and T2-relaxometry MRI. I have nothing to disclose. Cerebral small vessel disease is the most common cause of vascular cognitive impairment. And white matter hyperintensities are one of the most important hallmark MRI findings in VZI and relate to cognitive decline. Increases of local interstitial fluid and water content have been suggested to predate the cell loss in white matter hyperintensities. Therefore, the research question of this research is to gain more insight into the microstructural changes of the normal appearing white matter and the white matter hyperintensities in patients with VZI and controls. Our study sample comprises of 14 patients with VZI and 9 healthy controls. All patients underwent 3T MR imaging, and the scanning included a 3D T1 weighted image and a T2 flare. And to measure the interstitial fluid markers, we used a multi-echo Grace scan as well as a multi-B value diffusion weighted scan, for which the parameters are shown on this slide. I will now continue to explain how we derived the interstitial fluid markers, starting with the multi-B value acquisition. When looking at the MR signal with relatively high B values, we observed that the signal decays exponentially. However, the signal for lower B values does not follow this relation, due to the presence of an additional perfusion-related component. This is referred to as the IVIM effect. And from this, the perfusion fraction can be calculated. Traditionally, the diffusion and perfusion components are separated using bi-exponential modules. However, recently, spectral analysis using, for example, non-negative least squares gained a lot of attention. Using spectral analysis, we can obtain a spectrum of the IVIM components for a given voxel. For example, the spectrum of a voxel might look like this, with a microvascular perfusion component and a parenchymal diffusion component. From this, the perfusion fraction can be calculated. Looking at a different voxel, placed for example in a white matter hyperintensity lesion, the spectrum could look like this, where we see a similar spectrum, but now there is an additional peak in the spectrum, which relates to the interstitial fluid. And we can calculate the contribution of this peak, and we can do that for all the voxels, and then we obtain this interstitial fluid marker. Subsequently, when looking at the multi-echo MRI images, we observe that the decay is also exponentially. So we have a measured signal, but this measured signal comprises of several underlying water components. For example, the cellular water, some component related to the water trapped between the myelin sheets, and a free water component. And using spectral analysis, we can again separate these components and determine a free water fraction, as is depicted on the slide. So first of all, we try to see whether these two metrics related or that they are different or have complementary information. And what we found was that the free water fraction and the interstitial fluid fraction show a positive relation, but are not significant. So therefore, we conclude that they are indeed possess complementary information. When looking at group differences between the two study groups, we find a trend towards significance for a higher free water content in the VCI group compared to the healthy control group for both metrics. Next, we assess differences between normal appearing white matter lesions, and perilesional tissue. First, we observe that the intermediate fraction is significantly increased in perilesional white matter compared to the normal appearing white matter. Furthermore, for both markers, the white matter lesions exhibit higher water content compared to the normal appearing and perilesional white matter tissue. To conclude, this study provides indications for higher interstitial fluid, volume and mobility in the normal appearing white matter, of VCI patients compared to healthy controls. This increase might be an early marker for white matter hyperintensity development. Moreover, especially the ISF mobility, so the one that we assess with the multi-B value sequence, the intermediate component, seems to be an early marker for white matter hyperintensities as it is already elevated in perilesional tissue. So therefore, these quantitative ISF markers have the potential to visualize and monitor white matter hyperintensity development in vascular cognitive impairment. Thank you very much for your attention. If you have any questions regarding this work, I would be very happy to answer them.